My group at the University of Minnesota, we study particle dynamics, and part of work is on bacteria, which we treat them as moving particles. So I'm going to talk about our research on locomotion of flagellated bacteria in this talk. Before I get started, I should let you know who really did the work. So the first part of my work on the influence of a complex fluid on locomotion of bacteria is done by Xiang Cheng, my student at UMN. He graduates and then collaborate with Xiang Cheng at the Hainan University, China. And the second part of my talk, I'm going to talk about aerotaxis, how bacteria responds to oxygen, and that was done by my student Deep, Dipanjan Ghosh. I saw Deep just join the room, and also the theoretical part by Brito Chakrabarty from Tata Institute of India. So our research is funded by NSF and Parker Foundation. So the leading character of my story today is really E. coli. I think this is probably the most well-studied organism on Earth. All different aspects of this organism has been studied. To us, of course, we are most interested in how they move around, how they swim. So E. coli swim by rotating this long helical flagella. The swimming speed is about 10 to 20 micron per second. And because they are so small, the length is only about 3 micron, the width only 1 micron. The speed is so small. If you calculate Reynolds number of their swimming, the Reynolds number is on order of 10 to negative 5. So of course, this is really small, and it's in the world of in the fluid world where viscous effect dominant everything. It's quite different from our daily life experience on swimming at the high Reynolds number where inertia dominant. Although it's unfamiliar with to us, but certainly it's very important to understand how those micro swimmers move around. More specifically, how E. coli swim. So as I said, E. coli rotating this long flagella bundle. So this bundle is made of actually several or multiple flagella. So each flagella is driven by an individual separate motor, as I show here on the screen, by this tiny yellow part. So the motor is only 50 nanometer in width and rotating at 120 hertz. So it's really fast rotation. I cannot do that with my finger. So one time this motor rotates 120 times. And flagella grow randomly on the surface of the E. coli body, almost randomly on the surface of the body. And when all the motor rotate in the same direction, counterclockwise, if you look from behind, those flagella will form a bundle. And if one of the motor rotate in clockwise direction, the bundle will essentially just open up. That make the bacteria randomly tumble. So this gives you the classical run tumble motion of flagellated bacteria. So run times are on order of 1 second, and then one of the motor rotate in another direction, as I said, and the tumble lasts about 0.1 second. And then all the motor rotate in the same direction again, and they form a bundle, and the bacteria can move again. So this go back to the wrong place. Of course, the question why they do that, right? They don't waste energy to do this in vain. They can actually couple run tumble motion with the background chemical clue to allow them to do the chemotaxis, which I'm going to talk about in the second part of my talk. Another important thing that I'm going to use later in the first part of my talk is what's the geometry of this bundle. As I mentioned that all the flagella grow randomly on the surface of the body. You can imagine when those randomly grow flagella from a bundle, it's going to be very rare that the bundle axis line up with the body axis. So the slides that I show you here before, this is really idealized situation where the bundle perfectly align with the body. But most likely, as I show you here in this slide, they probably will form some angle. I call this angle alpha. So basically, the flagella pushing the body, but body sliding in some angle. Because of that, even in the wrong phase, the bacterial trajectory is not actually a straight line. It's actually forming this helical shape. I exaggerate here in the bottom figure here, but you can imagine when bacteria moving around, they doing this helical motion. And under microscope, because microscope is essentially a 2D projection of the 3D motion, you can see the body wobble back and forth. This is just a typical optical microscopy, bright field microscopy. You don't see flagella because they're very thin, very small in thickness. You don't see flagella, you only see the body. You can see when this guy moving around, it's actually wobble back and forth. This is because the bacteria actually swimming in helical trajectory. When you do 2D projections, become wobble. And if you look carefully for this bacteria coming from right, you can see this guy actually wobble so badly, almost like a gyrating moving forward. This is because the alpha angle here is close to 90 degrees. When you're pushing body this way, body actually is rotating, doesn't go forward too much. And this has been studied before, as I show here with this GFM paper. People document the trajectory that you can see from the body. So I'm going to use this effect later in my talk. Okay, so now let's move to the first part of my talk. I'm going to tell you how complex fluid influence the motion of bacteria, the swimming of bacteria. So the work is actually inspired by old work from my colleague Ed Kostler. He retired from our department right before pandemic. He asked this very interesting question: will humans swim faster or slower in syrup? Okay, so syrup is 2-3 times more viscous than water. Essentially, he asked if you have fluid has higher viscosity, increasing the background viscosity, will human has a faster swimming speed or lower swimming speed? And he decided run experiment. So he goes to University of Minnesota swimming pool, filling the whole swimming pool with syrup, in this case, guar gum solution. And he asked a professional university athlete, swimmer, to swim inside and document their swimming speed. In a typical in-person seminar, I will stop here, ask people vote on the results. I guess for online, I will not do that. But I will give you a couple of seconds to think about, do you think they swim faster or slower? Okay, so at the round experiment, when I give a talk, usually people will, or some people actually suggest that it depends on how you swim, right? So you may think that if you do butterfly, a lot of your body is outside the water, you may get less drag. If you do freestyle, your body is more inside the water, you get more drag. It depends on how you swim, you may have a different speed. Actually, Ed has all different swimming styles from butterfly, backstroke, freestyle. So no matter which style the athlete used, you can see the swimming speed doesn't change. So the plot here showing the relative speed normalized by swimming speed in water as function of basically the swimming speed. You can see they all line along one. So of course, in this case, as I mentioned early on, there's a human swimming, the size scale, the length scale is very large, swimming speed is high, Reynolds number tend to stay. Inertia really dominant the swimming behavior. And then someone read a very interesting article, news article in Nature, saying that had Ed Kostler done his experiment on swimming bacteria instead of humans, he would have recorded much lower time in syrup than in water. So the general idea actually is easy to understand. If you're thinking about the bacteria flagella motor, as I introduced early on, flagella motor has a constant motor output, right? So you have this characteristic line for motor performance, the red line, and then you have a load line, which depends on the viscosity of the fluid. When you rotate your motor, rotate your flagella, you will feel the load. And the intercept of the two, which is right out here, determine the rotation frequency of the motor. Now you're increasing background viscosity because inertia doesn't really matter. Increasing background viscosity, load line will increase. When you're increasing the slope of the load line, the rotation frequency of the motor goes down. And everything's linear at low Reynolds number, Stokes slow. When you have a lower rotation frequency, you have a lower swimming speed. So that makes sense that you expect the bacteria swim slower. Another analogy you can make is that when you bag a bicycle, when you have a constant motor, a torque on your pedal, but now you're going uphill because you have to have an extra load, then you have to battle slower. When you battle slower, you're going forward slower. So that's why you get slower speed. But apparently whoever read this article here didn't check the literature. Back to last century, 70 years ago, someone already studied when they put a polymer into fluid, which increasing the background viscosity, they measure all different type of flagella bacteria, whether they swim faster or slower. So very interestingly, when you add a little bit of polymer into a solution, increasing the viscosity a little bit, bacteria actually swim faster, as you can see from this line here. They plotted this plot as a bacteria swimming speed as function of 1 over viscosity called fluidity. So you can see from right to left, while increasing viscosity, there's always a peak for different type of flagella bacteria. Circle one is for E. coli. You can see the speed go up a little bit. And then when you add a lot of polymer in, of course, the solution becomes really gooey, and then they slow down. So you can see the decrease of speed at later time. And then following that work over the last 70 years, so 60, 70 years, there are many, many studies. I only list a sub-list of the paper here and some recent study. As you can see, all those studies show that when you're increasing viscosity of the solution, shear viscosity, the speed of bacteria swimming go up, speed of bacteria go up. Okay, so the reason why people study this so much is because this is certainly very relevant. Although our fluid mechanics class always tell us or teach us how bacteria swim in water, but in reality, in their natural habitat, very rarely they swim in water. Right? No matter that's in our body, in soil, in ocean, there are always different macromolecules, DNA, proteins, or different particles. They usually live in an environment which is non-Newtonian. So it's important to understand how they swim in those non-Newtonian environments. So that's why this sort of finding is really interesting and people study a lot. Of course, over 70 years, there must be a lot of theory to try to understand why that happens. So at least the four major theory here. The first theory is right after the first publication in 1970s. Howard Berg, if you know Berg, is a really established figure in the study of bacteria swimming. He basically proposed this idea that when you put a polymer into the solution, polymers start to form this gel network, and bacteria start to move inside this pore formed by this gel network. And then if you do some mathematical calculation, which is actually done by Kuto, 2002, you can see that you can get some swimming speed enhancement. And apparently this is not correct from polymer physics point of view, because if you put a polymer molecule into the solution, when they form in gel network, the pore size should be the size of, roughly the size of a radial gyration of the polymer coil, which is on order of a tenth of nanometer. And bacteria is 1 micron in thickness. There's no way that a bacteria can swim in any of those type of gel network. So clearly this is not right explanation. But of course, if you look at literature, because the status of Berg, this is still the most well-studied work explaining why bacteria swim faster in polymer solution. There's more recent work using polymer physics, I think, in a better way. So the idea is here. Because the bacterial flagella rotating at 120 hertz, as I mentioned earlier on, 120 hertz is really high shear rate, and the thickness of flagella is only order of a tenth of nanometer. So you're really creating a strong shear near the flagella. And we know polymer solution shear thinning. So if you're thinking about this geometry, you have very rapidly rotate flagella, which is shear thin of your polymer solution. The body is much bigger compared to flagella. To balance the torque, the body rotates at much slower speed, on order of 20 hertz. So the body feel much higher viscosity compared to flagella. The body feel the normal viscosity of polymer solution. Flagella shear thin the polymer solution feel smaller viscosity. So then you can have a two fluid around this scale of 1 micron. So you can build up what they call two fluid model, which by adjusting the parameter, you can see some speed enhancement. The third one is sort of along this line, but using different aspects of polymer physics. So because the shear rate is really high near the flagella, you can imagine when you have a polymer molecules near the flagella, you start to deform the configuration of the polymer, you start to stretch them out. When you stretch out the polymer solution or polymer molecules, you start to get normal stress difference called hoop stress, right? It's like a rod clamping of classical phenomena of polymer solution. And when you have this hoop stress, you can also have a speed enhancement. The last one is actually a very interesting one from Wilson Pong, Eisenberg. They did this measurement in 2014. They used the PVP from Dow Chemical, the original source of the research, and they dialyzed the polymer sample, get rid of small molecular weight contamination. They found the speed enhancement goes down a lot. And they argue that the reason why bacteria swim faster in this paper is because the bacteria eat small molecular weight contamination in the system, and they get more energy, they swim faster. So it's not a physical explanation, but more biological part. So this is the four major theory in the field try to explain what happens. Okay, so we go into the field. Of course, we don't want to go into the field and adding a new theory here. Well, we start to look into much simpler system. And then turn out that this much simpler system actually help us to understand why bacteria swim fast in polymer. I will talk about that. But what's our system? We study bacteria swimming hard sphere suspension. So really simple system. You just have small hard sphere colloidal particle. You add them in bacteria suspension. You ask how they swim. Do they swim faster or slower? Or they don't care about the presence of the particles. Experimentally, it's very simple. You just have a microfluid channel. We fill in our particle. They are more or less monodispersed particle. We use a polystyrene and PMMA particle, and the particle size range from 100 nanometer up to 1 micron. When changing the volume fraction from 0 to about 20%, this is really limiting by imaging. Go beyond 20%, scattering is so bad, we cannot do imaging. The size of the particle is limited by the sedimentation. If you have too big particles, they sediment to the bottom, you cannot have uniform suspension anymore. And then we just use wild type E. coli, and then we do imaging, see whether they swim faster or not. Here's the result. When I use a 100 nanometer PS particle polystyrene, this is the real size on the screen here, the size ratio between the bacteria and the particle. We see about 5% enhancement in speed at a volume fraction of particle about 2-3%. So very low volume fraction, you get tiny, small speed enhancement, about 5%. So I plot everything normalized by swimming speed in water. And then I increase in size of particle. When I go to 400 nanometer PMMA particle, in this case, I see about 50% enhancement at a volume fraction of about 3% or 4%. Okay, so this is pretty pronounced. The largest particle we use is 1 micron polystyrene particle. In this case, again, at about 4% volume fraction, we see about 80% enhancement. So this is quite a lot of enhancement. I always like to draw the analogy here. This is my student, Xiang Cheng. He's a good runner. This is his swimming speed compared to world champion swimming running speed. So 80% in human scale is huge. Actually, you can clearly see under microscope that bacteria swim faster when adding particle into the system. Okay, so now what I tell you, this is sort of a summary of what I tell you so far. If I'm plotting the maximal speed enhancement that I see as function of the size of particle, normalized by size of bacteria, which is always 1 micron, you can see the larger the size of particle, the faster the bacteria swim. Well, what's interesting is that you compare this with a polymer result. So we search all the polymer results where they use more or less uniform or low polydispersity polymer molecular weight. So if we using the hydrodynamic radius of polymer coil, we can actually collapse all the polymer results on top of our colloid result. So the color symbol here is from all different sources of a polymer study. And Wilson-Pong's results here, the orange one, you can see indeed compared to the original 1970s result, their speed enhancement goes down a lot because they purify the polymer better. Okay, but this is not the only similarity. So another interesting feature we see is that the swimming speed is anti-correlated with the wobble angle. So as you can see here from this plot, when the swimming speed goes up, the wobble angle goes down. When the swimming speed goes down, wobble angle goes up. I think this intuitively makes sense, right? If I wobble a lot, I do not go forward much, my swimming speed is low. If I swim more straight without much wobble, I get faster motion in the translational direction, I get higher swimming speed. So this all makes sense. If I plot this velocity as function of wobble angle, I see almost linear anti-correlation. But what's interesting is that if you compare this result with what reported in polymer literature, there are only 2 papers in polymer literature where they measure this angle. You can see that they also follow almost quantitatively between the 2 systems. Okay, again, this is not the only similarity. We also look into the feature of the trajectories. So on the left is bacteria swimming in water. So you can see the trajectory is short and zigzag. But as long as when we add particle in water, you can see the trajectory is longer and smooth. Same thing happens for polymer system. This is from Paula Risha at Penn. You can see that without polymer, in this case in water, you get zigzag and short trajectory. Their trajectory is a little bit longer than us because it should be same, right? This all bacteria in water. The reason is that they use some quasi 2-dimensional geometry, and we use a bulk, so that's why our trajectory is a little bit shorter. But feature is here. You can see that when they add a polymer into the system, the trajectory becomes longer and smooth. So very similar between the 2. Now, from the trajectory, we can extract a lot of different things. We can extract the rotational diffusivity, mean runtime, mean tumble time in the system. So I plot that as function of volume fraction particle, and in polymer system, they plotting this as function of the concentration of the polymer. And there are a lot of data in this slide, but the bottom line is that they all quantitatively similar between the 2 systems. So the conclusion here is that all the 7 reported swimming features are quantitatively similar in dilute colloid suspension and dilute polymer solution. So from this experimental observation, we make this conclusion that there must be some common underlying mechanism that make bacteria swim faster in both colloid suspension and polymer solutions. Okay, not mathematical proof, just based on the experimental observation. So if you believe this conclusion here, now we can use in our result to examine the existing theory in the field. So I just want to remind you, what we see here in colloid suspension is that with a volume fraction less than 4%, very low volume fraction, we see more than 80% speed enhancement in bacterial swimming speed. So first of all, at a full volume fraction there's no way particles touch each other forming any network. They're actually pretty far away from each other. So this networks idea cannot work. Shear thinning. So full volume fraction percent, this is a regime where even Einstein relation for suspension rheology or viscosity still work. So you just apply Einstein formula, you see that at a full volume fraction, the viscosity increase about 10%. Let's say you shear thinning so much, you get rid of completely the 10% actual viscosity. Essentially, you swim in water. It's still far from enough to explain the 80% enhancement you can have because shear thinning is so weak in this case. We use hard-shaped particles, polystyrene KMA. The modulus is 24 gigapascal. There's no way that bacteria can deform the particle, give you any normal stress difference in the system. There will be some normal stress difference due to Brownian motion of the particles, but that's really, really weak compared to polymer normal stress. So the normal stress difference cannot be the reason, at least in our system, to give you the speed enhancement. Of course, we wash our particle really well. We make sure there's no polymer contamination. There's no food for bacteria in this case. I was naive enough. I worry that the E. coli can eat polystyrene PMA. I asked my polymer friends whether that's possible. They tell me, Xiang, if this is the case, we don't worry about microplastic in the environment anymore, right? Everything will be degraded. So certainly this is not the reason why they swim faster. So basically we're saying that now the theory explains what will happen in our system. Now I give you some alternative explanation, some possibilities why that happens in our system, and hopefully also explain what happens in the polymer system. So let's imagine that you have a particle, you drag a particle with some force, let's say F to the right, and then the particle, of course, moving to the right. At low Reynolds number, this is just a Stokes law, 6 pi eta A, right? A is the radius of the particle. But now let's imagine that you have a particle moving next to a wall, which is stationary. Because the particle has to satisfy or the fluid has to satisfy no slip boundary condition at particle, at surface of particle, at surface of wall. So particle, even though you're only moving particle to the right, particle will automatically rotate. We call that boundary-induced torque. People know that, right? So now let's introduce this idea. Consider that when bacteria swim next to a particle. So when you have a bacteria swim next to a solid particle, the bacteria will feel an extra torque toward the particle due to this boundary-induced torque. And you can calculate that using lubrication theory, so which give you this formula here. So I just highlight several things. So first of all, gamma here is the size ratio. This is what we plot in our plot. R is the size of the particle normalized by size of bacteria. That's the gamma here. You see this logarithmic dependence because it's a lubrication approximation. The distance here is the distance between bacteria and the particle. And from the literature, we know the distance can go to very small. We use a 50 nanometer in this case. Okay, so we know bacteria swimming in water is a torque-free condition. This actual torque feel from the fluid must be balanced by something on bacteria. We argue that this actual torque is balanced by the torque given by the flagella hook. When flagella links to the body, there's a flexible flagella hook. And the hook has a natural state or natural angle, we call that theta 0. And then when you bend it, go to a new angle theta, you will feel actual torque there. So we assume the flagella hook is a linear spring, so we get this bending stiffness K, and this is theta is essentially the angle difference from the new angle and the equilibrium state angle. So now we just balance this two torque. We're assuming this torque-free condition. When you balance the torque, you basically get the relation between the wobble angle and the size of the particle, because the original angle here is the original wobble angle. The new theta will be the theta of the new wobble angle. So the first thing we do is we just take the experimental result, anti-correlation between wobble angle and velocity. Then you can link essentially velocity to the size ratio of the particle. This is the result here. And there's only one fitting parameter, which is the bending stiffness of the flagella hook. So we're using this formula to fit our results. It works really well. And the fitting gives us K equal to 7 times 10 to the negative 19 Newton meter. And Howard Berg has done this measurement a long time ago, 1989, using optical tweezer to measure the bending stiffness of the hook. They found the hook has the stiffness about 5 times 10 to the negative 19 Newton meter. So it's really same order of magnitude. Then of course, we should argue why this also applies for polymer. The reason why it's applied for polymer is because if you have a polymer coil in water, polymer actually behaves like a non-draining object, right? It's really in the ZM limit when you have a dilute polymer solution, meaning that when you have fluid passing through a polymer coil, it cannot penetrate into the coil. There will be a hydrodynamic radius. At the surface of hydrodynamic radius, you still have no slip boundary condition. Then all the argument that I have before can also apply in the polymer case. Of course, when you go to semi-dilute regime or concentrated regime, then they overlap with each other. You go into the draining case, that's Ross model for more concentrated polymer solution. But in our case, we're talking about dilute polymer solution. Non-draining is the good limit for what happens. The last thing I want to show you quickly here is we use this empirical relation between the wobble angle and the swimming speed, but can you essentially calculate the relation between wobble angle and swimming speed? That actually can be done. It's actually pretty easy in idea, but tedious in calculation because you need to consider about helical trajectory of bacteria, how you link the helical trajectory to the swimming speed forward. We did the calculation. I'm not showing you detail. Essentially, we just use a Purcell model, but extend the original classical Purcell model into 3 dimensions. We're thinking about the body and flagella forming some angle there, and from that, using force-free condition and torque-free condition, you can calculate actually swimming speed along the axis of the helical trajectory, which give you this solid line here. You can see this allow us to predict the swimming speed as function of wobble angle. So if you use that back to our original theory there, you don't need any fitting to get the speed enhancement. Okay, so let me quickly conclude the first part of what I have here. Essentially, I just tell you that when you add a polymer, add a colloid in your system, in the dilute limit, bacteria behave similar in colloid system and in polymer system. And this makes us to propose a new idea why bacteria swimming speed get enhanced in the dilute limit in both colloid suspension and in polymer solution. We'll also provide a new sort of more detailed calculation of bacterial wobbling that allow you to link the swimming speed to wobble angle. Okay, so I'm going to switch a gear a little bit. But if you have questions, certainly you can put your question on the chat box and I'll answer questions after the whole talk. The second part, so the first part is already published. The second one is what Deep is working on. So this is still the ongoing thing. We haven't really published a paper yet, but I want to tell you what we're working on right now in the lab. So the second part is about aerotaxis. So as I mentioned in the introduction, bacteria do the round and tumble motion because they want to couple this round and tumble to the background chemical clue in the environment. This allows them to move to the direction they want, usually chemo-attractant, away from the direction they don't want. Usually this is a place where you have a chemo repellent. Out of all the chemicals the bacteria sends through, oxygen really stand out due to multiple reasons. First of all, oxygen is really the chemical that first people found bacterial response to. Right? This is the beginning of chemotaxis research by Engelmann. He actually used this idea of bacteria loving oxygen to detect the most efficient part of a light spectrum, which is most efficient for photosynthesis. Okay, but this is also the first research showing that bacteria show chemotaxis behavior. And of course, oxygen is everywhere in microbial habitat and has a very strong influence on bacterial metabolism and also motility. That makes oxygen quite different from other chemicals. And in more recent years, in the context of active fluid or active matter, people also study how oxygen interacts with the bacterial suspension, showing all this very interesting collective patterns or emergent patterns. But in all those research, I should emphasize that people always treat oxygen as a standard chemoattractant. So bacteria just like to go to the location where there's a high concentration of oxygen. But if you look at the microbiology literature or biophysical literature, the response of bacteria to oxygen is actually quite controversial. It's very different from the typical chemoattractant that you can think about. Let me show you some literature results. So first result is from Adler. This is a classical paper on chemotaxis, probably one of the most cited paper on chemotaxis, where basically he put bacteria in this capillary, almost one-dimensional channel, and let E. coli to migrate. So as you can see here, after 6 hours, bacteria gradually forming a band first, and this band gradually moving to the interface. at interface with the air. So based on this result, basically they argue bacteria like options like chemoattractant allow bacteria to chemotaxi and migrating to the surface. But there's some relatively speaking, more recent work by Liebschaber. They published the PR work 2009, almost similar geometry. But what they found is the opposite. As you can see here, as time goes on, the opening, the interface is at x equals 0 on the left, but bacteria moving away from interface. So sort of like bacteria really hate oxygen. They're going away from oxygen. And in between 1997, there's some PNS work where people show that if you put bacteria in this capillary channel, bacteria actually forming a band. The location of that is not at interface, not in the bulk, away from the interface, really at some intermediate location. And based on that, they argue that oxygen, actually bacteria sense optimal level of oxygen. This is called optimal sensing. So like balance of hate and love. So the first question, of course, we want to ask here is that how could the same bacteria in a similar geometries show qualitatively different migration behavior? What happens? There must be something that's not well controlled in all those experiments. Why one case migrating to the surface, another case migrating to the bulk, and third one just stay in the middle. And next one is sort of in the context of the active matter. If optimal oxygen sensing is the mechanism for E. coli sense oxygen, how does optimal sensing determine the population dynamics? Because all the existing studies really treat oxygen as chemoattractant. If we treat oxygen as an optimal sensing chemical, what is the new dynamics we can have in the system? So we start again, we like a simple experiment. We just start with a very simple experiment. So in this case, again, capillary channel. We just have 42-dimensional channels, and we open the channel from one end at x equals 0 on the left and close the channel on the right. So this is basically dead end. Oxygen can only diffuse from left to the right. And then we mix both fluorescent labeled bacteria and non-fluorescent bacteria. So this will allow us to see bacterial motion at even higher concentration because we only see subfraction of a bacteria, small fraction of a bacteria. So each of bacteria in this picture, specific picture you see here, for each of the bright bacteria here, you see there's on average 4 bacteria that you don't see. They are dark, they are not fluorescent labeled. So then we just looking bacterial trajectory as well as counting number of bacteria at different locations. There's 3 different colors just indicate different location in the channel. As you can already see from this picture that in the middle of the channel, there's a higher concentration of bacteria. So we then measure the dynamics of bacterial migration as function of time for different overall bacterial density, so average bacterial density. When the average bacterial density at 1 times 10 to 9 cells per milliliter, you can see bacteria start to form a band at an early time. So you can see some small peak here. But as time goes on, this peak starts to migrating away from the interfaces at x equals 0. So it seems that for this concentration, bacteria hate oxygen. They want to migrating away from oxygen. When I go to higher mean bacterial concentration, 2 times 10 to the 9, you can see that they also form a band at short time, but the band location stay. So in this case, bacteria like balance of hate and love, they do not migrating to the interface or stay or migrating away from the interface. They stay at the location where they form a band. Now, if I go to high concentration, 4 times 10 to the 9 cells per milliliter, you can see a short time form a band, but as time goes on, the band starts migrating toward the interface. So in this case, they do love oxygen. So now you can see that actually the controversial result or conflicting result is really because there's a different total concentration of bacteria. At low concentration, they're migrating away from the oxygen, and a high concentration migrating toward the oxygen. At the intermediate concentration, they stay where they are. To understand this better, so overall bacterial density is the control parameter here. To understand this better, we build this 2-dimensional kinetic theory, which is a very simple equation here. So basically what you have is just write the probability density function of bacteria. So what is the probability found bacteria at location x, at the orientation theta, at time t? And then you write this, essentially the conservation of the probability. And the yellow one is the translational dynamics, how the translation changing the probability. The blue one here is rotational diffusivity. And this pink one here is because bacteria tumble. When they tumble, they're moving out of this angle. And the last one is tumble in. When from another angle they tumble, they can go into this location. And if you integrate out the orientation component of degree of freedom, you get the number density of bacteria at different locations. And then the most important thing here for this theory, make the theory fit our experiment, is we're assuming there's an optimal oxygen sensing. So we set up this green region. When the oxygen concentration in this green region, bacteria tumble is just a normal tumble. When bacteria moving away from this green region, they will tumble toward the green region. So for example, if you're in the red region, if bacteria moving to the right, they will extend their run time, so they will tumble less. But if they're moving away from the green region to the left, they will go back to their normal tumble behavior. Same for the blue one. If they're moving to the green, they will extend their run time, they will reduce the tumble, but when they're moving away from the green region, they will go back to their tumble behavior. And then we couple this with the normal diffusion consumption equation for oxygen. C here is the concentration of the oxygen. Okay. So when you have this simple kinetic theory, you run your simulation. So what I'm going to show you now here is the bacterial density in the channel from 0 all the way to the end of channel. And the blue curve here is going to be the oxygen concentration inside channel. As you can see, when time goes on, first you do see a band forming, meaning that bacteria forming concentrate at a certain location, and they can see gradually this band start to migrating. So if I get the peak of this band, I'm plotting that as function of time. I can see that when density is low, below a certain density we call UC, rho C, you can see the band location keep migrating away from interface into the bulk. If I increasing the concentration at rho C, you can see at beginning there's some migration, but once the system stabilizes, the position of the peak does not change anymore, so stay there. If I go to higher concentration after transient period, the peak position of the bacterial band migrating toward the interface. So you really see the three different behavior that we have in the experiment. We can be more quantitative. So we extract the migration speed from the kinetic theory, which is the right one. So they follow the same trend as the experiment, although there's a factor of 3 difference. But I should emphasize that in our kinetic theory, there's really no fitting parameter. All the biologically relevant parameters from our measurement, independent measurement, or from literature. So there's no fitting parameter. We can get speeds follow the same trend within factor of 3 of the experimental result. And more importantly, you can see the speed across 0, meaning that bacterial migration direction flip at this critical concentration rho C. So that gave us a prediction of rho C. There are 2 key components to get this migration. The first component I already mentioned is the optimal oxygen sensing. So you basically want the migration, the tumble frequency of bacteria on both sides of the optimal oxygen concentration flip. So lambda minus here is the tumble frequency of bacteria moving toward the interface. Lambda plus here is the tumble frequency of bacteria moving away from the interface. If you are on the left of the optimal oxygen concentration, lambda minus is bigger than lambda plus, because bacteria want to migrating towards optimal oxygen location, then this delta lambda is bigger than 0. If on the other side of this optimal concentration C star, this sign is a flip, because now bacteria like to migrating toward the interface. So delta lambda become negative. So you must have this sign change in the difference of lambda in order to get the bacterial band. Because theoretically, you can show, I don't have time to show the detail, the bacterial concentration is actually follow the integration of this tumble frequency difference. So because tumble frequency difference goes through change sign, you'll form a peak at location of C star. So you get a peak formation. In the sense that this is not surprising, right? Once you say that there's an optimal concentration of oxygen, bacteria will just move to there and form a peak. It must be the case, right? So this is logically very easy to think about, intuitively very easy to understand. What is more complicated here is then why the peak migrate. That's a little bit more non-trivial, right? So to have a peak migrating, you must have separation time scales between the fast oxygen diffusion consumption and slow aerotaxis response of bacteria. So we can show that by doing this simple calculation. Assuming there's a time separation, first you set up your bacterial concentration uniformly at this average concentration rho 0. You can calculate just using the oxygen consumption diffusion equation to look where is the optimal concentration of oxygen. Now from that, you can back up the peak location. That's basically XP0 I show you here with this black line. So this is under assumption that the bacteria does not move, even though all the bacteria consume oxygen. Next, you can argue that when bacteria start to move, because at a longer time, aerotaxis become important, you can again calculate the location of the optimal oxygen concentration and then associate the peak position, which is the star symbol that I show you here. In both cases, they all decreasing as function of the overall concentration of bacteria. Sorry, this should be U0 here. But you can see there's actually two different regimes. The two lines cross at this critical concentration. On the left of this critical concentration, the star is above the solid line. That means that the final location of the peak is longer or larger than the initial position of the peak. So bacteria peak migrating into the bulk. On the right of this row C, the trend is opposite. The end location is on the smaller side or the left side of the initial position. So the migration direction of bacteria is to the interface. At row C, they don't move anymore. So that's where the slippage happens. That gives you the peak migration. In the last 1 minute, I want to show you some result that we want to convince you indeed there's the optimal oxygen sensing, because this is a sort of new thing, at least in the active matter community, that we want to convince you that for E. coli, they do sense optimal concentration of oxygen. So we do two things. First, we measure single bacterial dynamics to show you that their migration direction actually different on different side of the peak. So we calculate, or we measure the run length of bacteria on the left of the peak, on the right of the peak. As you can see, on the left of the peak, the run length is longer when bacteria moving to the peak. The run length is shorter when they are moving away from the peak. The trend is the opposite on the other side of the peak. If you're on the right side of the peak, when you're moving to the peak, you get longer. And when you're moving away from the peak, the run length is shorter. From the run length, again, you can calculate the tumble frequency. The average tumble frequency will basically just spinning speed divided by the run length. And from that, you can get lambda plus more than lambda minus on this side. Lambda plus is bigger than lambda minus on the right side of the peak. That gives you the sign change that you really need for optimal oxygen sensing. And more interestingly, if you just calculate the magnitude of this change of lambda, it's actually balance the density gradient that you see in the system. This is telling you that the peak that you see in your system is really due to this difference in tumble frequency, which is sustained this peak we have in our measurement. The last thing is that we're also looking into the molecular signaling pathway, how this optimal sensing be achieved in the system. We're looking into different mutants. This is AER or TSR. This is just two receptor of a bacteria. If you knock out this receptor, you can see they're still sensing the optimal position of the oxygen, which essentially you can think this is similar to thermotaxis E. coli perform, because for thermotaxis, they're also sensing the optimal position where you have the right temperature for them to live. And then if you're using the signal pathway divided by our UHI2 group, just mapping the thermotaxi idea into aerotaxi, you can actually predict the location of peak from kinetic theory. This is y equal to x1. Again, I don't go to the detail. I will list the bioRxiv paper. If you're interested, you can certainly look into the theory in more detail. So let me conclude here. So in the second part of my talk, I tell you how the aerotaxis modify the swimming behavior of bacteria. We look into this problem from both molecular signaling pathway, motility of single cell, as well as collective population dynamics. So our result resolves some long-time controversy about where the bacteria goes, right? The migration direction of bacteria in self-generated oxygen gradient. We identify an overall bacterial density as a control parameter. At this critical bacterial density, essentially you do not see migration anymore. This may be useful for bacteria to transform from motile state into a sun cell state that allows them to form biofilm. And we show some evidence of optimal sensing from single bacterial level. And we think that this will open up some interesting idea to use optimal oxygen sensing assumption to study the emergent behavior in active matter. So there's some archive paper we post there. So with that, I will end my talk here, see if there are any questions from the chat. Thank you.